Hi everyone, I am Maria Fernanda Rodríguez from third semester, and today I'm going to talk about the immunomodulatory effect of microglia on experimental cerebral malarial neuroinflammation via the PD-1 PD-L1 pathway. First, we need to clarify some concepts, so let's start talking about neuroinflammation. It is an inflammatory response within the brain or spinal cord, and it is mediated by cytokines, chemokines, and secondary messengers, which are produced by microglia and astrocytes. It can lead to recruitment of immune cells and potentially cell death. Next, let's talk about the PD-1 PD-L1 pathway. PD-1 is an inhibitory immune checkpoint which is expressed on the surface of T cells. PD-1 pathway plays a subtle role in maintaining peripheral T lymphocyte tolerance and regulating inflammation. So, when we activate it, we block and reduce the damage that can result of the intense activity of T cells. The objective of this article is to elucidate the negative regulation mechanism in the experimental cerebral malarial brain mediated by microglia. Furthermore, to investigate the protective effect of the appropriate enhancement of the PD-1 PD-L1 pathway in the brain against experimental cerebral malarium through the intrathecal injection of the adenovirus expressing PD-L1 IgG1 Fc fusion protein. Let's continue with some of the methods that were used on this project. On qPCR, a fluorescent reporter dye is used as an indirect measure of the amount of nucleic acid present during each amplification cycle. The increase in fluorescent signal is directly proportional to that amount. It was used on this research to check cytokine levels and the increasement of PD-1 and PD-L1 levels. On flow cytometry, we have three main components: fluidics, optics, and electronics. The fluidic system is responsible for transporting sample from the sample tube to the flow cell. Optical system include excitation light sources, lenses, and filters used to collect and move light around the instrument, and the detection system that generates the photocurrent. The electronics are like the brain of the flow cytometer. Here, the photocurrent from the detector is digitized and processed to be saved for subsequent analysis. It was used to track positive cells for PD-1 over the days. Immunofluorescence staining is a technique that uses the specificity of antibodies with fluorescent dyes to recognize their antigen, and therefore allows visualization of the distribution of the target molecule through fluorescent dyes with a fluorescence microscope. It was used to localize stones that expressed PD-1 and PD-L1 over the days. On reverse transcription PCR, the principle is to convert RNA into its complementary DNA sequence by reverse transcriptase, to synthesize a second strand with DNA polymerase, and finally to generate a complementary DNA molecule which can be amplified by PCR in the normal way. Now let's check the results. As we can see on this electrophoresis, which is the picture number 1, protein levels of PD-L1 and PD-1 in the brain were more expressed 6-7 days post-infection. Also, according to immunofluorescence staining, which is the second picture, there are like some changes of brain tissue sections. Positive cells for PD-1 and PD-L1 were widely observed in the cerebrum, cerebellum, and olfactory bulb of experimental cerebral malaria mice on the 7th day. So, according to this, it means that the infection induced the PD-1 PD-L1 pathway in the brain. Here we have another electrophoresis yields, which shows on picture number 1 that the protein level of PD-L1 increased significantly in the brainstem and the cerebellum, while even though we cannot see it clearly on this picture. According to the authors, a significant increase in PD-1 protein was detected only in the cerebrum on the 7th day post-infection. Finally, let's analyze the picture number 2. This one demonstrates that immunofluorescence, that the expression of PD-1 and PD-L1 in the brainstem was observed on the 7th day after infection. According to both techniques, the changes in PD-1 and PD-L1 were different in each region of the brain. Now let's compare what the article says with the statements made by other authors. According to Swanson, peripheral blood PD-L1 IgG1Fc interference inhibited the activation of cytotoxicity of CD8 positive cells, and this statement agrees with the investigation of this article. Bowlby says that microglia played an important role in the pathogenesis of inflammation, demyelination, and nerve cell injury, and this is also one of the main points of this article. According to Hamanaka, microglia subgroups have functional differences or even have special immune regulatory functions, and this is also in agreement with what is said in the article. In conclusion, we can comment that first of all, molecular biology techniques are very important to discover new forms of reducing the damage of serious illness that can affect the human being, and secondly, that research and investigation are essential to make progress in the world of medicine, being excellent ways to reach effective and lasting solutions. Thank you.